and thank you guys for inviting us to present our data. Obviously, it's been a long time in coming to get to a level that we present. You know, we were working on this before Alberto Ascherio's paper. And so just to say that we've been trying to figure out how viruses cause autoimmune disease and what its role as an essential cofactor is. I think that's what we believe, not that it's the initial cause or anything like that. But our data in mouse models using a virus similar to EBV has demonstrated that just the act of being latent is enough to immune modulate towards a disease like MS. And then Jessica has spent her PhD career in my lab defining this in a humanized mouse where we can actually use have the effect of actual EBV to show how it can immune modulate a disease like EAE to look more like MS. So this is really an essential function of the virus's ability to do this. And Jessica's incredibly talented, technically driven person who has figured out how to give EAE to humanized mice of multiple genotypes, and she's going to do that, present it today to you. And this is our work that hopefully will be published shortly. Thank you. Here's Jessica.
Okay, thanks everybody. So I'm going to talk to you guys about what I have on the slide here, which is the finding ultimately for our humanized mouse model that Epstein-Barr virus infection promotes a Th1 mediated disease in humanized mouse and a humanized EAE model. Here we go. Okay, so I'm sure we're all somewhat aware of what MS is, but I want to draw your attention specifically to the cause of MS. And I know that the cause of MS is actually not entirely known, nor is it well understood, but it's thought to be due to a combination of both environmental and genetic factors. And one of those environmental factors that Marc's lab is really focused on is viral infections. And so we end up seeing the most strong and consistent evidence linking infection to MS through Epstein-Barr virus. Many people know Epstein-Barr virus as a cause of infectious mononucleosis. Usually when you're a young adult, you get a sore throat for a while, you're very tired, but mostly it goes pretty unrecognized, it isn't highly reported. However, there's a really strong geographic correlation between cases of mono and cases of MS. And on the right here is actually an earlier study from the Asscherio group at Harvard, where they show that if you've had mono before, you're at a higher risk for developing MS. Whereas if you asymptomatically acquire EBV, you're at a moderate risk. And if you've never had EBV, it's very unlikely that you'll have MS. So it's not only just that you have the virus, but that you've had an immune response to the virus that dictates your risk for MS. We also see in individuals who go on to develop MS that they have higher antibody levels towards EBV, and they're usually up to diagnosis. And of course, as was brought up earlier, the 20-year-long US military study by the Asscherio group that was published in Science last year showed that EBV seroconversion confers a 32-fold increased risk for developing MS. And that this was specific to EBV and not to CMV, which is a related herpes virus that infects the immune compartment of people. They also showed that seroconversion with EBV specifically precedes the appearance of a axonal degeneration marker in the blood. So it's happening before you have evidence of damage in the brain. And another sort of clue as to how EBV could be actively playing a role, not only in the onset of MS, but during its progression, is that B-cell depletion therapy is very effective for inflammatory forms of MS. And we know that EBV is a gamma herpes virus that latently infects B cells and can be targeted by these therapies, at least in animal models. The problem with studying EBV infection in an animal system or in a disease model is that it doesn't infect rodents, and rodents are typically used to study MS through EAE or Cooper's models, whatnot. It can very inefficiently infect some rabbits, but for the most part, doesn't mimic the full spectrum of disease for both the infection or MS. So in Marc's lab, the group has taken a twofold approach to assess this relationship between EBV infection and MS. The first that he touched on briefly was that we model the virus to suit the mouse. In this case, we use gamma herpes virus 68. It's a natural rodent pathogen with very many similarities to both Epstein-Barr virus and Kaposi sarcoma herpes virus. And it also establishes a latent infection in mouse B cells. And the second was what I'm going to talk to you about was what I was doing for my PhD project in his lab, was to model the mouse to suit the virus, which is to engraft a human immune system within these mice, which are then susceptible to infection with EBV directly. So this is the first approach, again, the model the virus using gamma herpes virus. So this is a homologue of EBV. It behaves very similar in its latent form to Epstein-Barr virus. And what we do is we infect, we infect regular C57 black 6 mice, which is the typical mouse strain used to model MS. Wait 5 weeks for the virus to establish a latent infection, so it's no longer actively replicating. And then we induce EAE using the typical MOG 3555 epitope emulsified in CFA with pertussis toxin. And this is considered a pretty standard autoimmune model of MS to direct that T cell response to the CNS. And what we see is that gamma herpes virus infected mice that are given EAE have an immunopathology that reflects MS more closely than uninfected, naive EAE mice that we use to generally understand how MS works and test our therapies. And this includes more brain involvement. So there's a lot more CD8 positive T cells that enter the brain. We see more of an intense skewing toward the Th1 response and downregulated Th17, as well as general peripheral and local suppression of Treg functions. And interestingly, this is only seen with the latent infection. If you give it a virus that's unable to establish latent infection, only has an acute phase, it is not able to enhance the disease the same way as the latent infection does. And we don't see this with the murine version of cytomegalovirus, which also establishes latency in a different cell type. It seems to be a very specific effect to latent infection within the mouse. B cells, yeah. Okay, and then the second approach here is to model the mouse to suit the virus, which is using humanized mice. So humanized mice, in our case, don't necessarily refer to genetic humanization or transgenes. What we mostly mean is the engraftment of human immune system components into immune deficient mice. There's a few ways you can do this. You can transfer mature human blood cells, which is what I'm going to be speaking about, but you can also engraft hematopoietic tissues, stem cells, in order to reconstitute the mice with whichever subsets you're looking for. So this is the design that we use to perform our experiments. We first obtained 3 groups of blood donors, the first being people who have an existing diagnosis of MS, all of which were EBV seropositive, as you might expect. And then we also had a second group of individuals who have never had a history of autoimmune disease that are EBV seropositive. And then our third group were people who have no autoimmunity and have also never been exposed to EBV based on their serology. We then isolate total peripheral blood mononuclear sites from their blood samples. We inject those into NSG mice, which are NOD-SCID gamma. These mice are severely immune compromised. They can't make their own T cells or B cells, and they also do not form functional NK cells. So they engraft quite efficiently with human donor cells. Following 3 weeks, we allow the T cells mainly to reconstitute into these mice, and then we check their blood samples to ensure that the human CD45 positive cells have actually engrafted. So we have an effector T cell population to work with to induce EAE. And then we induce EAE using a combination of both full-length MOG protein, the extracellular domain 120, as well as the MOG 3555 epitope. And we do this just to provide as many MOG epitopes as possible for the different T cell specificities. For our donors, we included individuals who were female between the ages of 20 and 40. For the RMS diagnosis, these individuals had to be completely treatment naive throughout their life, have a short disease duration because we are trying to look for that earlier immune modulation that occurs early in disease, and a low disability score because, again, a higher disability score could suggest advancement of the disease. And we have here just the specifics for each individual, which you can see on the drop site link if you're more interested. We then checked out their EBV status. And so not unexpectedly, we saw that our individuals with MS had the highest EBV IgG responses to both. Can you guys see my cursor if I do this? Yeah. Okay. So they had more intense responses to the viral capsid antigen. So this is considered an acute phase antigen, as well as to EBNA1, which is the latency MS-associated protein to maintain the episome. And the responses are greater in our people who have MS compared to people who've acquired this virus naturally, but have not developed MS afterward. And again, we just confirmed that our EBV seronegative donors don't have any response to either of these antigens. We then did just a general qPCR to assess viral loads in the peripheral blood and nuclear cells of these donors to confirm that they weren't experiencing any EBV-associated condition that would suggest a confounding factor such as mono, a chronic active infection, reactivation, et cetera. So we're looking at people with latent EBV infection. We then transferred those cells into the mice, and then we induced EAE. And so for those who are not as familiar with EAE, EAE works by introducing a specified antigen, in this case we use MOG, in the context of an immune adjuvant. We use complete forage adjuvant, and as well as we also administer pertussis toxin, which is thought to breach the blood-brain barrier to allow those autoreactive T cells to migrate into the CNS. And the way that you score the disease severity is based on the extent and localization of the paralysis. So typically paralysis will start at the tip of the tail and ascend upward into the hind limbs and forelimbs. And so what you can see in A here is that mice who receive PBMCs from people who have an existing MS diagnosis experience a significantly overall greater disease burden compared to mice that were derived from individuals who had EBV but didn't have MS. And then we can see here that individuals who had never had EBV or MS had this delayed onset of their disease, and their severity was quite a bit lower than here. What I find interesting is that anyone who's familiar with EAE models in general might know that NOD mice, they don't have that classic wave of symptoms where they stay paralyzed until the end of the experiment. NOD mice have this relapsing phenotype that's very similar to what you see for RRMS. And we see that with these mice as well. And we can actually see that our EBV seropositive groups, regardless of MS, onset at a very similar time, but that our MS patient mice continue to get sicker while our healthy donors sort of come back down to the level of an EBV negative. So our EBV positive healthy group looks like an intermediate between the two, negative and MS positive group. We can see this trend of increasing disease severity also by disease incidence. So we have 30%-ish overall disease incidence that is delayed in our EBV negative mice. It onsets pretty similarly with our EBV positive groups regardless of MS, but then our MS groups sort of outpace the EBV positive healthy group. And we also see this again, if we quantify just by symptom onset day, our EBV negative mice have a significantly delayed onset of EAE symptoms compared to the EBV positive groups. As far as other risk factors, we also normalized to see if we had any heightened response to CMV, which again is a related herpes virus, but is not considered to be negatively associated with MS outcomes. We did see that our EBV positive RMS group did have a slightly elevated CMV level compared to other groups, but on the whole, the difference wasn't highly significant to suggest that our effect could be attributed to CMV. We also checked for existing antibodies towards Rh-MOG in order to rule out individuals who might have MOG-associated antibody disease. And we saw that none of our donors had significant reactivity to the inducing antigen. And then we also checked serum vitamin D levels. And at the time that these individuals donated, they all had very similar vitamin D levels on the whole, which again suggests that these factors are probably not what's explaining the differences we're seeing clinically. We also know that when immune cells enter the CNS, that they mediate demyelination of the spinal cord. And we wanted to see if the increased disease severity that we were seeing in our MS and EBV positive groups was due to increased demyelination of the spinal cord. And so we ended up sectioning the entire way along the spinal cord and doing an overall summation of the degree of demyelination. And we saw that here's our NSG controls. These are mice that are not humanized, and they have a full demyelinated spinal cord. And that our EBV negative mice are protected from significant demyelination as compared to our EBV positive groups, as well as these are NOD control mice. So these are a classical EAE model. So they're better protected from demyelination when EAE is induced compared to our EBV positive groups. And just to highlight some of the differences that can come up when you look at MS immunopathology versus EAE. MS is typically considered a brain dominant disease. It does affect the spinal cord in some cases, but EAE is very spinal cord dominant. You tend to see a lot of CD4 T cells infiltrating the spinal cord, whereas in MS, CD8 T cells tend to be more abundant within the lesions of the brain, or at least on the outside, and anywhere from 2:1 to 10:1 CD8 to CD4. And you can see a mix of both Th1 and Th17 in both MS and EAE, depending on the model you use in the case of EAE and how you induce the predisposition of the strain. And on the right here, I just want to draw your attention to what we see in this model. So we see both CD4s and CD8s entering the brain, which suggests that, we're having a model that's experiencing immunopathology, so MS, that the brain is involved. Oops, sorry. Oh, my mistake. We can see this now. Sorry. I zoomed out accidentally. Human CD4s and human CD8s. Most of our human CD4s that are entering the CNS are highly interferon gamma producing. Those that produce IL-17 actually tend to be double positive. So you see relatively few single positive Th17s. And then associated, we have these CD8 T cells that are highly cytotoxic. So a lot of them express either interferon gamma and or granzyme B. We also determined that if you look, so this is for the same group of mice, so mice that are humanized with the same donor PBMCs. If you classify the mice based on whether or not they got symptoms, you can actually see which immune cell types contributed to the production of those symptoms in those mice compared to ones that didn't get symptoms. So in A here, we saw that in the brain and spinal cord specifically, we had a higher CD8 to CD4 T cell ratio in the mice that developed symptoms as compared to those that remained subclinical to the end. There was a slight elevation in the spleen, but not as significantly as the CNS, which suggests that we have an MS-like disease happening that's involving CD8 T cell infiltration into the CNS. And if you look at just the total human immune cell count in the brain, there's not a huge difference between the total cell counts between mice that did or didn't get symptoms, but in the spinal cord there is. And so the number of human immune cells that actually enters into the spinal cord is a determinant of whether or not these mice develop symptoms. And specifically, it's how many human T cells. This is CD3. And so EA symptom grading in our model reflects spinal cord pathology due to human immune cell infiltration. And specifically, we can see that this, I don't think I had the slide here, but we don't actually see a numerical difference in human CD4 T cells or activated human CD4 T cells numerically between mice that do or don't get symptoms, but we do see this is the case for CD8s. So specifically, the more CD8s that are able to enter the spinal cord, the more likely a mouse is to get EAE symptoms, which suggests that we have a disease process that models what we think might be happening in MS. And that also then raises the question of, well, then how is demyelination mediated in a system where you have human immune cells in mouse tissues that are not necessarily genetically compatible? So the first thing we looked at was murine macrophage counts. So NSG mice, we don't, in this model, irradiate them in order to clear out the existing innate immunity. We leave that intact. And what we can see is that in the spinal cord specifically, there are far more murine macrophages in the spinal cord compared to mice, between mice that did or didn't get symptoms. So there's much more murine macrophage involvement. And then if you look at which proportion of these infiltrating murine macrophages actually contain myelin, indicative of their phagocytic capacity, it's much higher in this, as expected, it's higher in the spinal cord and brain than you would expect in the periphery where there is no myelin. And that this number of macrophages that have phagocytized myelin correlates very strongly and specifically with the number of human CD8 T cells that has entered the spinal cord, which again suggests that human CD8 T cells are entering into the spinal cord, they're producing cytotoxic molecules, promoting damage in the CNS, and that the murine macrophages in this case are either directly mediating phagocytosis of myelin or they're cleaning up the debris. And we know that this is also something that happens in MS lesions. So this is a paper from 2015 where they looked at CD8 T cell infiltration into MS lesions. And they generally saw that there were more CD8 T cells compared to CD4s in the brain and spinal cord, and that they also produced cytotoxic molecules like granzyme B. So in general, this characterization data of our model suggests that we're at least seeing similar T cell pathology that you would expect to see in MS, and that we could use this model to study how EBV affects that T cell response in this model. We also took a closer look at the spinal cord. Here on the left, we have an NSG mouse that was non-grafted with immune cells. You can see the myelin staining in green. NISL is a neuron marker, or RNA, DAPI, and human CD8, so these yellow cells here. And in mice that we give PBMCs to and induce EAE, there is a specific recruitment of human CD8 T cells into the area where there is this loss of green myelin staining. And then we also see this in the cerebellum. So on the left, here's a control NSG mouse. You see this compact, neatly organized myelin structure, whereas it becomes a little more frayed and dull. And then you can see these human CD8 T cells entering into the area. And what's interesting is our flow data had suggested that they cooperated with myelin phagocytosing murine macrophages. And we can actually see them co-localizing together within the cerebellum. So this white staining here is IBO1, which is considered a murine microglia and macrophage marker. And they co-localize to areas where these human CD8s are appearing, which further suggests that there's an interaction occurring here where our human T cells are mediating damage to the CNS, and it's either being cleaned up or affected by the presence of these murine macrophages as well. So just to summarize the immunopathology of this model, why we ended up looking at certain outcomes we did, we see that human immune cells demyelinate the CNS of NSG mice when they're engrafted with PBMCs and induced with log EAE. We see that there's infiltrates composed of both human CD8 and CD4 T cells, that this seems to be very strongly Th1 and TC1 driven, and that there is myelin phagocytosis by the murine myeloid cells that appears to be driving the demyelination that we're seeing. So getting back to our question of, well, which cell type is actually causing the difference we see clinically between these groups? The automatic question is, well, is it B cells? EBV is a B cell tropic virus. And in general, something to note about PBMC engrafted humanized mice is that B cells do not tend to survive very well. They get outcompeted by T cell engraftment. And so we generally see very few B cells engrafting. Within the spinal cord, we saw between 25 and 100 total B cells compared to, 10,000 to 1 million T cells entering those same tissues. And most of our B cells were actually in the spleen. So the presence of B cells within the CNS that were or were not infected didn't seem particularly likely. So we checked out the spleen, and we saw that except for one lone mouse here, we didn't seem to transfer any EBV infected B cells directly into the mice. And that they didn't have any reactivation of memory B cells towards viral capsid antigen that would suggest that an EBV specific infection had been transferred into our recipient mice. And that corresponds with our data in the donors where we didn't see viral loads that were detectable in their blood at the time of donation. But what we did see was that EBV infection history really affected the degree to which the T cells responded in these mice, even though the virus itself wasn't brought over with the T cells. So in the case of CD4 T cells, we can see this stepwise increase in the number of human CD4 T cells that enters the spinal cord, and a much higher number in the brain of MS-derived mice compared to our EBV negative mice. So it suggests there's EBV is a factor that increases the number of CD4 T cells. And then again, having MS and EBV is another risk factor for that increased inflammation in the spinal cord. In the brain, we also see that, with human CD8 T cells, we also see in the brain that our MS patient-derived mice have more CD8s in their brain, and that both EBV positive groups have slightly more than our EBV negative group. And this difference in terms of the number of cells that actually enters into the CNS between mice that have or have not received PBMCs from people with MS or EBV is highlighted when you express these T cell counts as a ratio of the number of regulatory C cells in those same tissues. So on the C here, we have interferon gamma producing as well as FOXP3 expressing T cell ratios. And again, there's this pretty clear stepwise increase in all three tissues, brain, spinal cord, and spleen, that shows that a history of MS and EBV increases the rate at which interferon gamma CD4 T cells are present compared to Tregs, suggesting increased inflammation is because of reduced regulation compared to increased effector. We also see this trend with human CD8 T cells that express interferon gamma and granzyme B. So the cytotoxic compared to regulatory T cell ratio also increases when you add EBV and when you add a history of MS. What we also find interesting about this finding is that if you look at the gamma herpes virus model that I mentioned earlier, when you latently infect regular black 6 mice, these are not humanized mice, gamma herpes virus, and then you induce with the same antigens, you see this same skew where our infected mice have a higher CD8 to CD4 T cell ratio in the brain, spinal cord, and spleen. They also have an increased Th1 to Treg ratio in the brain, spinal cord, and spleen compared to naive mice, as well as a TC1 ratio. So this suggests that gamma herpes virus infection in its latent form in itself is a predisposing factor to having a more aggressive Th1 TC1 response compared to a subdued regulatory response. And that isn't necessarily specific to Epstein-Barr virus in humans, but also just to gamma herpes virus infection in an immune system generally. And we wanted to look more specifically at when exactly this Treg deficiency begins. So in our pre-engraftment blood donor PBMC samples, we can see that there's no significant difference between the donor groups. So we had 4 RMS EBV positive donors, 4 EBV positive healthy donors, and 3 EBV negative donors. The proportion of their human CD4 T cells expressing FOXP3 was very similar between the groups. But as soon as you engraft these mice, you see this stepwise stratification again. So it suggests that even just the non-specific reactivity of these T cells to be engrafted into a host with mouse tissues is enough to show this reduced Treg expansion when you have an existing EBV infection, and even more so when you have an EBV infection and a diagnosis of MS. And then when you induce EAE, this reduced Treg expansion translates into reduced Treg expansion in the CNS, which we ultimately believe drives the more intense disease clinically that we see. And we think that this general Treg deficiency that we've seen with our other models, as well as in this model, could explain how EBV is also linked to other autoimmune diseases if it has this more broad immunomodulatory effect of suppressing Treg expansion upon challenge. So specifically in terms of RA and lupus, we think EBV could be driving a similar response. And we also have model data in the black 6s with RA showing that happens as well. And then we also wanted to see, so separate from the Treg deficiency, does EBV promote T cell proliferation of any specificity? Is this a specific response? In general, we've seen that EBV or gamma herpes virus infection of black 6 mice, when you induce them with MOG EAE, doesn't lead to a specific expansion of MOG specific T cells in those mice. So we weren't expecting the MOG specific T cells to necessarily be expanded specifically by EBV. But what we saw is if you actually just take PBMCs from your donors that have or haven't had EBV or MS, and you stimulate them with CD3, CD28 beads, so this is just general TCR stimulation without any specific antigen, this T cells that are derived from people who have had EBV, even compared to just people who haven't, they proliferate much more strongly in response to the stimulation. And again, perhaps not surprising, we also see this with our individuals who have MS. But I do think it's really interesting that, it may not be surprising that MS patient-derived T cells expand more readily upon challenge, but this is a difference we're seeing just between people who have or haven't had EBV. And we also see this effect with our human CD8 T cells as well. So it suggests that EBV is this priming factor for an increased T cell response that's less regulated and more effector-based when you've had a latent infection. And so that's where we're at now for this model. We have some interesting stuff coming down the pike, but as far as significance of what we've seen so far, we've shown that EBV status and MS diagnosis, both independently and together, exacerbate the clinical and immunological disease severity that we see in a humanized mouse model of MS, and that EBV appears to do so not through direct infection of these tissues or reactivation, but through altering the balance of effector and regulatory T cells that actually infiltrate the CNS once an autoimmune challenge has been administered. And more generally, we're glad to have developed this more personalized preclinical model that we can hopefully use more broadly to make personalized mice based on the individual donor status to identify mechanisms, biomarkers, and assess therapies. And with that, we'd like to thank all of our funding sponsors, so UBC, both the MS Society of Canada and the USA, Canadian Institutes of Health Research, as well as all kinds of collaborators who have helped us do various aspects in the lab, and of course, Marc. Yeah, so happy to take any questions and discuss. Oh, you think you're muted, Mike?
I was going to say, you can put questions in the chat. And I can see that John Corboy already has a question.
No, that, would you like me to read the question?
Can you see it?
Yeah, I can read the question out and then we can chat about it. Now that we have a potential vaccine for MS, is there concern that whatever happens with EBV infection being associated with MS would be similarly seen as part of a vaccine response to MS? So could a vaccine induce the similar response that we see as actually having the infection? Marc, would you like to have first stab at this question, or I can as well?
Well, I think our data shows that it's the action of maintaining latency that is driving immune modulation, not actually the infection or the immune response, direct immune response to the virus. So a vaccine to EBV would in itself not necessarily induce MS any more than a vaccine to any other virus in that way. But it's actually the way that chronic viruses appear is because they have reached a détente with viruses, with the immune system. And so the immune system is actually changing itself to accommodate this chronic infection. The virus is also changing itself, but we're less interested in some of that. But it's changed itself to maintain latency. But it's really the action of the immune system that's now immune modulated and thinks that anything coming down the pike might be EBV, which it needs a nice strong Th1 response to. So that's kind of our feeling of what the mechanism really is here.
Yeah, so that balance, is it because of I think there's been a lot of work to suggest that there's impairment of inducible Treg within multiple sclerosis, and you could hypothesize that a viral infection might do that. Do you see that in these animals?
Yeah, so in our mouse model, where it's just mice, we see that as soon as you establish latency, the Treg numbers are dropped down, and it's really a strong driver of why we see pathogenic disease later on. So in that model, you can, because it's mice, you can actually test a lot of different things. You can test induction of arthritis, induction of lupus. You can infect with other viruses, bacteria, and everything that the response is a strong Th1 response with a very reduced Treg response. And so I think that possibly is what's happening for people, and likely a real strong component of it.
Did you look at functional Tregs at all? Did you do functional assays?
We have
Just
not.
looking at numbers.
Not in the human
Right. And
example.
so you know
in
that
the gamma
activated
herpes
cell,
virus, we have looked at function. It seems to be just a generalized Treg reduction numbers, not necessarily function.
right? Because that's what we found in MS actually.
What's that, Steve?
Functionally, we have a lot of problem seeing functional defects in Tregs in MS patients.
In our mouse model, where it's black 6 mice, that's exactly what we see.
Well, I was going to
It's a
say,
number thing, and you can't get your numbers up.
No, I like the number, but I think because activated cells have lots of Treg markers on them, right?
Yeah.
So to us, I think unless you do a TSDR or some other molecular approach to actually look at the methylation or the function,
Yeah,
I think
we haven't done that yet.
right.
yeah, I was going to say, there's a couple of things. One is, it depends how you look at it, because, there was work from, I believe it was Nitin Karandikar, showing that almost all human T cells as a part of an activation express FOXP3. And so you can't just look at FOXP3 as defining that. But I can tell you that if you stimulate to try to induce Tregs with TGF beta, MS patients then are impaired compared to healthy individuals. Does that,
Right,
are
and
there,
they're huge. I think we're all back now.
Yeah.
There are huge differences between mice, mouse immune cells and mouse Tregs than human Tregs and how
Yeah.
they function, and that whole breadth of what cells express FOXP3.
and
So-
then in your model, you're always using adjuvant immunization, right?
Mhm.
Yes, I can tell you that we have, we don't think that EAE is an MS in any way, shape, or form, but
Of
it gives us
course.
a nice little test, right? So we have actually, and I can't really tell you what now because we haven't published any of it, but we have other ways to induce similar responses, at least in the mouse model and a little bit in the humanized mouse model, but they all involve creating inflammation. So we have to create inflammation and we have to have antigen to our target.
Mhm.
But they don't have to be EAE as described.
So, we've done very similar work in the non-human primate, right? In the marmoset,
Mhm.
PNAS paper, right? With another herpes virus. Very, very similar. And there, so EAE in that model looks great, actually, from a human, because we're MRI centric. So we put the monkeys in the MR, we see lots of lesions, so we could follow them. But we also looked at the upregulation of virus in our system.
Mm-hmm.
Okay. So I will tell you again, even on one of your first slides, Jessica,
Mhm.
when you found, I'm sorry, we're getting a lot of helicopters in Washington. That you found no latent virus in the MS patients, right?
Not at the level that it was detected to
Right.
suggest
We
that we
can
had
detect that.
symptoms.
We can detect that in MS patients. So
Mhm.
in fact, we find about, well, it depends if you're relapsing remitting patients, are in the active phase or are they active at all at the time?
No,
you know
they
if
were
they're
all in remission.
They're in remission. You should also look at the active phase as well, because you will see a lot more virus. So we can detect
Yeah.
about 40% in PBMCs, EBV in stable MS patients, and which ups to about 80% in CD19 cells from the active disease. So even in the stable phase, there's actually a fair amount of virus in there, but we have different technologies.
Yeah, I was going to say, I think
No.
the method we had used was really just a quick screen to see if they had symptomatic EBV infection. That would suggest
Yeah.
like
And again,
more-
I'm just also curious. I've got to tell you, I've been doing this a long time. I've never seen an EBV negative person.
We found 4.
We found 4.
There must be something in Canada. I don't know what you guys are doing, but
I'm
That's
EBV
terrible.
positive.
I don't know anybody that has been. I mean, that was the thing with Alberto. He had all those EBV negatives, which was great, but we've never seen one actually.
Oh,
Yeah, we're lucky to have them.
That's amazing.
And then we just we keep
I,
them around. Yeah.
they're in a
So
little
when
box.
you did that in the mice, I saw the total number of mice, but how many of the individuals equal how many of the mice?
Right.
right?
So each person can make 12 to 20 mice from their donation
12 to 2.
based
So
on
you
the
had
number of
36 mice, say, from the EBV negative people? You had
Yep.
36 mice.
cells.
Wow, that's a lot of mice. That is.
But
Have
they're very variably reconstituted, however. So that's another
Right.
factor. It's
Yeah,
not like
because
a
that
black
clearly
box.
is a subset of all the mice you had, right?
Yeah. Yeah.
Right. So do you do this as a score of how many work and how many don't work?
For symptom incidence.
Yeah, no, for the whole EBV story. I mean, because they're going to reconstitute at very different rates. How do you
Right.
normalize that,
So for this study, I specifically did not exclude any mice based on their
Okay.
reconstitution levels or on their symptom production, because I wanted to demonstrate that variability. And even with that variability, you still see this pattern emerging, which suggests it's a pretty strong
Yeah.
relationship if you can have so many differences even between your cohorts from the same person.
Yeah,
You
yeah,
still see this effect
the
on that.
variability among patients is a big deal, actually. It's a big factor. And to tell you, Mark, we're also, again, working in the marmosets. So we found that the marmosets actually carry an endogenous gamma liptocryptovirus,
Mhm.
LHV3. So we've been following that, in about 20% of our colony is actually infected. Well, in fact, 20% of all colonies are infected. You just no one looked. So there's a lot of a bit, and then the idea: could we induce EAE in those animals, like we did in the beta herpes virus study? You know, because we could do experiments like that. So again, I think these are, as you said, I think these are maybe common mechanisms among herpes viruses. They may not be specific to EBV or even MS, actually, in that case. But I think you're looking at a mechanistic approach, which is, as a virologist, that's even more interesting, frankly, because there might be something more common about that. And I really like that. It's a very nice model system. Have you thought of doing some single cell RNA work from the brains? You got the money? Come on,
we're trying. We need more money.
MS Society, they got a ton of money. Because
right.
so we're doing a lot of that now on cerebrospinal fluid and blood of 20 or 30 MS patients compared to controls. So that is really being a really important, and of course you could do that in real time from the brains of your animals, and then you really get the complete. Because one thing I think, as you said, Jessica, it would sure be nice to think that there may be some antigen specificity driven by those T cells, right? Are they recognizing EBV or something like that? Either with a tetramer, or if you could do TCR, we're doing TCR repertoire analyses and things like that. So there are high throughput type of technologies that are available. But again, there's a million things to do. So
there's absolutely a million things. And
yeah, it's great. No, really nice.
But it's exciting because there's lots to do and lots of ways to go here.
Oops, sorry.
Yeah, so for an old EAE person, I guess one of the things I would say, and I don't know, I mean, whether you've looked at other antigens, right? Because one of the things that Mark, Jessica, and I were talking about before is that now I remember the first papers that came out looking at MOG 35 to 55 mice, right? And the issue was they said, oh, this is in fact a great model for neuromyelitis optica, when in fact it was probably a great model for MOG associated demyelinating disease, right? And so in the old days, people, when they were inducing EAE in NOD mice, I mean, Steve Miller was using a PLP epitope. I think Sami Apori was as well. We always use the MVP TCR transgenic from Joan Government. And I can tell you that those mice, if you induce EAE, they get the sort of typical, if you're looking at the spinal cord, there's just tons of lesions. But if you let them get spontaneous EAE, back in the day, the NIH mouse colony wasn't as clean as
I'm losing them.
You get mice that were sick. They'd have optic nerve disease. They'd have.
I'm losing you, Mike. I don't hear you.
Your volume's cutting out.
Yeah. Sorry about that. But anyway, so there's lots of factors that affect things like where the lesions are located when you start throwing in adjuvants and doing all this other stuff, as opposed to just letting the virus do the work for you. And I think that's what really probably happens in humans, is that the gamma herpes virus in both mice and humans affects the Tregs in such a way that now I'm able to get a much more robust Th1 response. My personal bias is MS is predominantly Th1, but if you want to study Th17 demyelinating disease, you should look at NMO and aquaporin 4.
Well, two bits of information that I can give you about the mouse model that Justin covers. One is that from the beginning, we decided, do we really need this pertussis toxin addition to inducing EAE? And it turns out we don't. A lot of time we still use it because we're superstitious, and we want it to work, and we don't want to chance it, and we have it lying around. But we don't really need it. The virus seems to allow us to bypass that. The other one that's really important in terms of the antigen is that we can do this in collagen-induced arthritis and show the same enhancement of disease. Now, we haven't gone and done that in a humanized mouse yet, but we can do it in the Black 6. And then we have tested other models of EAE, and Jess can speak to that a little bit more in terms of the antigens that we've used. But in almost, I think, every one that we've done, we see an enhancement. It's extremely profound. We were really lucky to choose the Black 6 MOG as our first attempt many years ago, but because it's so profound, the difference that we see. But we do see this enhancement in every other type of EAE that we've used or tested with.
But I can tell you, for example, with MOG 35 to 55, you can induce EAE in B6 mice that are deficient in B cells. And
Yep.
so when you're now putting in the immune cells, if I have human macrophages and T cells and I use a peptide, I don't need B cells, and you're putting in I assume you're immunizing with like 200 micrograms of MOG peptide, or
We do
somewhere in
peptide,
that
but we
range.
also do protein in order to activate that B cell compartment if we can.
Right. My point is that's overwhelming compared to what human T cells see under normal conditions, right? That's why the reason that B cells are great, because they have the B cell receptor, they concentrate antigen. Macrophages don't concentrate antigen, but if I put a ton of antigen in, then I don't need to concentrate it. It just picks up, so.
Mhm.
But these are all things, that are very interesting and what makes all these different models so much fun.
Yeah. Yeah, definitely talking about the molecular mechanisms by which we induce EAE, the fact that the peptide skews automatically towards the CD4 response just because of the structure of it.
With the monkey, we've used brain homogenate,
Mhm.
whole brain homogenate, actually. Because in fact, we think the MOG was a little too aggressive. Okay, in the monkey it was, actually. So we went to whole brain. In fact, I think that was a Mike Racke protocol that we're using.
There you go.
That we had. So can I make one maybe last comment to Bob Morris? So my very good friend, Alberto Escari, and I disagree on a few things. And I'm glad at least I think the way Mark and Jessica look at it, to go from the cause to maybe one of these environmental triggers, which is something I think I feel more strongly about. But we also have done very similar work to the science paper that we published. In fact, it's not simply just EBV. We can find other viruses as well in using the exact same platform. And in other diseases, we also can show other antigen-specific diseases. We also show the involvement of EBV. So it's not quite the slam dunk, at least that I think it is, all right? And maybe some others as well. But I know the way epidemiologists think of things are
Well,
a little different than not all of us have access to the DoD database of
right.
30,000 samples, right?
I mean, it kind of comes back to what you were saying earlier about how hard it is to find people who aren't EBV positive, and and obviously that there's no way they could have done that study in any lesser data set. But and it's so hard to study that question, and the fact that they were able to pull together the data to do it, and that the data showed those kinds of results. I mean, a 32-fold increase in risk associated with being seropositive. That's, you don't.
That was really
see that
based
in
on,
epidemiology.
it was only based on 30 patients, actually, when they started bringing it down. All right. But you're right. I mean, we always see increased antibodies to EBV in MS. That's known for many years. But we use the same platform because I think you're looking either for virus discovery or viral signatures. So there's an idea that's going on in these complicated diseases. It's not just the response to one virus or one antigen. It could be your genetics that allow you to respond to multiple viruses and not respond to other viruses that may give you this susceptibility signature. And again, at what compartment you look at, whether it's blood or cerebrospinal fluid. So we do a lot of work in CSF, actually.
Yeah, I mean, absolutely. I didn't mean to suggest at all that this was, the cause and.
Oh,
you know,
no, Alberto suggests that, and Gavin Giovannoni
Well,
completely suggests that, all right?
yeah, I mean, my suggestion, I just think it's, you know, you have to have that present. It seems to me, what their research suggests is you have to have it present. And then genetics come into play and environmental factors and all these other things, but without this sort of.
Right. So as much as I'd like to think we're conservative, and I'd like to think we're going ahead with an antiviral EBV drug in MS.
Right.
In a clinical trial.
Right. I mean,
we're ready to move forward in this. All right. But I think it's the work of having these animal models that I think are pretty important, particularly when you could humanize and you could do 32. That's actually a big. That's really impressive. I remember the LA colonies, they all got contaminated and the whole colony died. And it's really impressive, actually. You guys are doing that. So that's really great. The idea of personalizing, because I could tell you we are isolating EBV from patients and controls.
Mm-hmm.
EBV itself may be different.
Right. There may be a change in
a change, right? So what you're looking at is a different EBV or constituents when you do sequencing in the entire.
You mean or not like PML that's different in the kidney versus in the brain?
So, I mean, we're just looking at it we're making what's called a spontaneous lymphoblastoid cell. You make LCL cell lines with B95-8, right? You take any B cell, you infect it with B, but you're infecting it. Actually, that's a marmoset cell line, right? So you could take B cells from a patient, and if you're really good and you know how to garden, eventually you could get a B cell from that patient that contains its own EBV. Right? I can make one from Mark, and I can make one from Jessica, make one from Mike, and I can make it from 20 MS patients. And we could start comparing the two. So the idea there may be different, that's kind of on the virology side of it, actually. And then what is the immune response to that may be?
Yeah, I would be really interested in seeing beyond the LCLs is how the immune system responds to those viruses, just because.
We'd be happy to give you those cell lines if you like.
Perfect. Yeah,
Great.
I mean.
But they'll grow. I mean, but they're from the patients themselves. So we have it from patients that are in their stable phase, patients in their active phase, and healthy donors. Okay.
Mm-hmm. Yeah, I think Mark and I were discussing. Paul Lieberman presented LCL work.
Yeah, we did that with Paul.
Yeah, in Italy. We were chatting about this as well, and we were curious if that was a cause or a consequence of having MS, like if you have different outgrowth of EBV variants, if you could get your hands on people before they have their diagnosis, would you see the repertoire being different?
Well, that's the Ascario set. He could get people who are
exactly.
Says that, well, it's before their clinical onset. That doesn't mean they have something before that.
Mm-hmm.
But I am involved, going back to one of the questions, the National Multiple Sclerosis Society is having a panel discussion on should we be vaccinating children to prevent against EBV, to prevent MS. And so there's a meeting on that. And
I mean, it seems like there's enough other reasons to vaccinate against EBV that, it's hard to make a case that we shouldn't.
Well, there evolutionarily, it might be okay to have multiple infections that 99% of us do well with, right?
Theoretically, EBV, the philosophical question is, we've had EBV with us for thousands of years.
Yep.
And we've evolved to this state where we are now. And it's only been the last 100 years or so that these diseases have really popped up that are EBV driven. And it's because how we are transmitting EBV to people from person to person has changed in a dramatic way, because parents are no longer chewing food and they're sharing their saliva with their children. Instead, we're preparing our food for them separately. And so it's cleaner, but it's creating a situation where we don't get EBV till a later age, which is a big caveat of Alberto's data, because he's only looking at military age individuals. He's not looking at people outside that age older, and he's not looking at people that are children. Children catch EBV still, the epidemiology of it is that they are protected a bit more than when you get it at a teenage year.
And
So
the DoD beta is predominantly male too.
Yeah.
And
also,
MS is, 3 to 1, 2 to 1, 3 to 1 female. disease.
Female.
and there's
Right.
the other part of his, just on his data set is there's not a lot of mononucleosis data on there. So he can't really tell who had mono or who didn't.
Right. So this
Yeah.
goes back to the idea where, and again, you started this, Bob, saying, I'm not
Yeah,
comfortable
this
with the
one.
word cause, but it clearly could be one of the triggers, if not a very important trigger in
Mhm.
subsets of patients.
Yes.
Right. So it doesn't mean it's one EBV is the cause of, or the trigger for every patient.
Mhm.
And maybe through this personalized kind of way, we can subdivide the patients to say who
Mhm.
might be better for a therapy or a vaccine or something. But again, you're looking at the sledgehammer. We're all trying to find the virus. I always said HIV screwed it up for everybody, because if you got HIV, everyone got AIDS, right? And very few people get MS, and they all have EBV. Question is why? And it still may be subset analysis and different viruses as well. And again, we see this in many other chronic neurologic diseases. There's an interest in dementias and Alzheimer's and some Parkinson's. And so this could be more of a universal kind of an approach versus this is the MS agent,
Well,
That's my little spiel.
Yeah.
I really hate to cut this off. And in fact, we can keep the line open as long as you guys want to talk, but I know that some people do need to jump off.
Oh,
There
here.
is, when you do sign off, there is a survey just to get your reaction to this event and suggest your thoughts on future events that we could do. So please answer that. And we're actually the other thing of note is we're expanding our MS Research Exchange into other areas of neurology. So we're launching neuroexchange.net this month. We'll send the information around about that, but look for it. So thanks to the presenters. That was fascinating. Thanks to the commenters. That was equally fascinating. And I really, that was a great session. I really appreciated it. So
Thank
Thanks.
you all.
Well, thank you guys. I really enjoyed that
Bye-bye.
too.
Say hi to Amy for me, Mike. Take care. Bye-bye.
Yeah, bye.